Have you ever paused and wondered what's happening at the microscopic level in your body right now? Your cells are constantly at work, dividing, growing, and maintaining the very structure of your existence. This process, known as the cell cycle, is fundamental to life, and today we're going to dissect it in detail. Whether you're an AP biology student or just curious about how life sustains itself, this video will walk you through the fascinating world of cellular division, checkpoints, and what happens when this intricate process goes wrong. So without further ado, let's dive right in. At its core, the cell cycle is a process through which cells grow and divide, making it the cornerstone of life. But why is it so important? Well, without the cell cycle, organisms couldn't grow, repair damage, or even reproduce. Every cut on your skin that heals and every inch that you grow taller as a child happens because of the cell cycle. Think of the cell cycle as a highly coordinated assembly line. Each step must occur in a precise order to ensure the correct outcome. Two healthy daughter cells. But this process is more than just mechanical. It's a masterpiece of regulation. Any errors can ripple throughout the body, affecting tissues, organs, and even entire systems. For example, errors in cell division may lead to cancer, where cells can lose control and divide uncontrollably. So, understanding the cycle is not just a cool biology lesson, it's key to understanding diseases and how we might combat them. When we think of the cell cycle, it's easy to picture mitosis, the dramatic separation of chromosomes. But most of the action actually happens in interphase, the preparatory phase where the cell does the heavy lifting to get ready for division. Let's break it down step by step. Beginning with G1 phase, this phase is all about growth. The cell is like a construction site, bustling with activity as it increases in size and synthesizes new organelles. Importantly, this is where the cell prepares the materials it'll need later. But it's not just about getting bigger. There's also a checkpoint here. The cell inspects itself to ensure it has enough resources and that the DNA is intact. If something's off, it halts progress and initiates repairs. If the damage is beyond repair, the cell may trigger apoptosis or program cell death to prevent problems from spreading. Moving on to S phase, S stands for synthesis. Here's where things get intense. The cell copies its DNA, ensuring every chromosome is duplicated. This duplication is crucial because during mitosis, the genetic material must be split evenly between the two daughter cells. Think of it as creating a backup of your hard drive. Each copy must be exact down to the last byte. Any errors here can lead to mutations, which, if unchecked, may have serious consequences. Lastly comes G2 phase. In this phase, the cell continues to grow and prepare for division. It synthesizes proteins and organelles needed for the big event, mitosis. This phase is also home to the G2 checkpoint, where the cell ensures all DNA has been accurately replicated. If there's an issue, such as incomplete DNA synthesis, the cell pauses to fix it. This final quality control step is vital to prevent errors during division. After all the preparations, the cell enters the M phase, which includes both mitosis and cytokinesis. Mitosis is the process of nuclear division, broken into four key stages. The first stage is prophase. Within this, chromosomes condense, becoming visible under a microscope as distinct X-shaped structures. Spindle fibers, which are tiny structures that help move chromosomes, start to form. The nuclear envelope dissolves, marking the cell's commitment to division. The second stage is metaphase, in which chromosomes align at the metaphase plate, a central plane within the cell. This alignment is critical for equal distribution of genetic material. Special proteins called kinetochores attach chromosomes to spindle fibers, ensuring they'll be pulled apart correctly. Third phase is anaphase, in which sister chromatids, which are identical copies of a chromosome, are separated and pulled to opposite ends of the cell. This step ensures each daughter cell will have an identical set of chromosomes. The fourth phase is telophase, Within this phase, the chromosomes reach the cell poles and nuclear envelopes form around them. At this stage, the cell starts to resemble two separate entities. Finally, cytokinesis divides the cytoplasm, resulting in two distinct daughter cells. In animal cells, this involves the formation of a cleavage furrow, while in plant cells, a cell plate forms due to the rigid cell wall. The cell cycle operates with military precision thanks to a series of checkpoints. G1 checkpoint ensures the cell is ready to replicate its DNA. G2 checkpoint verifies the accuracy of DNA replication and checks for resources needed for mitosis. The M checkpoint, occurring during metaphase, ensures all chromosomes are properly attached to spindle fibers and are aligned for equal separation. If a cell fails a checkpoint, it may pause to repair damage, or if the damage is too severe, undergo apoptosis. These checkpoints are like traffic lights, ensuring the cycle progresses only when it's safe to do so. Cancer occurs when the cell cycle's regulatory mechanisms break down. Mutations in key genes such as tumor suppressor genes or proto-oncogenes can disrupt the balance. For instance, tumor suppressor genes act like brakes, stopping the cycle if something's wrong. Mutations here are like cutting the brakes on a car. Proto-oncogenes promote cell growth. When mutated, they can become oncogenes, pushing cells to divide uncontrollably. Unchecked, these cells form tumors, which can invade nearby tissues or spread through the body in a process called metastasis. At the molecular level, the cell cycle is regulated by proteins called cyclins and enzymes called cyclin-dependent kinases, also known as CDKs. Cyclins bind to CDKs, activating them to drive the cycle forward. Each phase relies on a specific cyclin CDK complex, ensuring tight control and precise timing. Some cells, like neurons, exit the cycle in the G0 phase, a resting phase where they function without dividing. For some, like liver cells, this is temporary. For others, like many brain cells, it's permanent. In conclusion, the cell cycle is a masterpiece of biological engineering, ensuring life continues seamlessly at the microscopic level. Its regulation highlights the balance between both growth and control, a balance that, when disrupted, can have devastating consequences. If you found this breakdown helpful, please give this video a like and share with your classmates, and subscribe for more deep dives into AP biology topics. Thanks for watching, and remember to keep it pushing and stay curious.